Good morning, everybody. My name is Virginia Kaklamani from UT Health San Antonio. I'm one of the co-directors for SABCS. Thank you for being here on our last day. This is the most difficult session to put together for our speakers. Trying to summarize a whole year's achievements into 30 minutes, I think is very hard. And when I did it myself, I know I worked the whole year for that talk. But it's also, to me, the most rewarding. So I appreciate you all being here. And if you miss anything, obviously we'll have it online in a few days. So to start out, so as you all know, we're going to have a basic, a translational, early breast cancer, and metastatic talk. And to start us out is going to be Dr. Andrei Goga from UCSF, Helen Diller Family Comprehensive Cancer Center. Professor Goga is a physician scientist, basic investigator, as well as a practicing breast medical oncologist, really a very rare individual. And he will be talking to us about the basic science achievements of the year. Dr. Goga.
All right, well, thank you very much, and it's a pleasure to present the basic science overview for the past year. I have no disclosures, and really in the last year or so, in the November to October period, there have been about 20 to 30,000 articles if you look at breast cancer and PubMed, and just between you and I have to tell you I haven't read all of them, but what I'd like to do today is to focus on those that were thought-provoking, impactful, and perhaps paradigm-changing. And so I would challenge you for each of the manuscripts. There's a PubMed ID. So if you know, I can't do any individual study justice. So if you're interested, please go back and read the original articles. And if you're really interested, please reach out to the the researchers, because I'm sure they would like to interact and hear from you. All right, so to break it down into a few different topics, we're going to talk a little bit about breast cancer subtypes, then technologies, immune oncology, and then finally metastases. These are I think the four areas that there's been the most progress in. So first question, the way I'm going to present this is in terms of questions. The first question, how to overcome CDK4/6 inhibitor resistance. We've heard a lot of talks at this meeting about CDK4/6 inhibitors. They're really the mainstay of treatment for hormone receptor positive disease. Unfortunately, most patients progress after a period of months to years. And just very briefly, many of these resistant mechanisms have already been described. Essentially, losing the control checkpoint with CDK4 and 6 can occur through many mechanisms, including upregulation of other cyclins, loss of RB, loss of other growth inhibitory pathway. And the net effect is that cells can now inappropriately enter the cell cycle. And sort of to step back a sec, when we think of the cell cycle, CDK4/6 inhibitors function here at sort of this decision point of whether the cell is going to go through the remainder of the cell cycle, enter S phase, and then go through mitosis. But it's just one of multiple cyclin-dependent kinases, and there's downstream cyclin-dependent kinases. So one rational approach is if you've lost this control pathway that's regulated by CDK4/6 inhibitors is to look downstream at another cell cycle kinase. And so just want to highlight a couple studies. One is using models that were derived to be resistant, in this case to palbociclib. You can now add a drug, in this case called dinaciclib, that hits multiple downstream CDKs, so CDK1 and CDK2, and this was shown to have activity. This seems like a very rational approach, and there was a whole session about targeting other CDKs. You can see that these resistant cells now underwent cell death. A second approach is to explore what's different about these cells. So you can imagine if you blow through the G1S checkpoint, then you may have defects in the rest of the cell cycle. And so, in fact, that's what was observed. So this is a separate study that explored cells that were made resistant to CDK4/6 inhibitors. And what was noticed is that they have very frequent mitotic defects, or they're inappropriately entering mitosis. This group did a screen of various drugs and identified a number of different mitotic kinases. So basically, again, hitting the cell cycle later with an inhibitor of a mitotic kinase called TTK or Aurora kinase B or Aurora kinase A. And so these had quite a bit of activity. So in summary, CDK4/6 inhibitor resistance is an active area of investigation. There's existing drugs, including drugs that hit downstream, such as CDK1, 2, 5, 9 inhibitors that can overcome some forms of CDK4/6 resistance, and also targeting further downstream in the cell cycle in mitosis might also be a useful therapeutic approach. So the next question is, what is the effect of HER2-directed HER2-directed antibodies on kinase activity? This has been an area of some controversy really for decades. Antibodies such as Herceptin and pertuzumab were designed to target the HER2-HER3 complex, in particular HER2, but it's really been unclear whether these actually inhibit activity or mainly are a way to stimulate the immune response and have antibody-dependent cell cytotoxicity. And so I think sort of to challenge this notion that they actually have any inhibitory effects, this is a study that was published which did a very detailed structure-function analysis and showed that when you overexpress HER2, even in the presence of pertuzumab, you really don't inhibit signaling downstream of HER3. That's not to say these drugs clearly work, but it challenges the notion that they actually inhibit the kinase. So the next is, so if these are acting mostly in some sort of immune way, what happens targeting HER2 low or heterogeneous tumors? And I think in this space there were several interesting papers. So just to put in perspective, we know now that breast cancer is quite heterogeneous. Most breast cancers are not comprised of just one type of cell, and you've heard about this from various advanced omics approaches, single-cell sequencing, and so forth. But what does that mean in terms of HER2-positive disease? So there was this very interesting neoadjuvant study that looked at neoadjuvant TDM1 plus pertuzumab in patients who were HER2 positive. And specifically, the study looked at the heterogeneity. So each patient had 2 biopsies, and their tumor was labeled as heterogeneous if at least either if there was one biopsy was negative, or if one of the 2 biopsies by FISH was between 5 and 50% amplification. And so the interesting observation was that if you were not heterogeneous, if both biopsies showed high HER2, then the path CR was about 55%. But if these criteria, which defined heterogeneity, then none of the patients had a pathologic complete response. So again, this suggests that the amount of HER2 and the percentage of cells is actually quite important, especially for drugs such as TDM1 and pertuzumab. I'm not going to go into it in detail, but I think it's been discussed at this meeting quite a bit, and this is a nice review that newer drugs such as trastuzumab, deruxtecan, may have some properties in terms of their linker, their payload, and their being non-polar, which may allow them to also target some non-HER2 high cells. So I think this is still an active area of investigation. It remains to be seen whether we're actually going to see better responses in early disease. That may in fact not be the case. So summary, HER2-directed antibodies have limited ability to block HER2-HER3 signaling in the context of HER2 overexpression. However, heterogeneity is important and is associated with diminished response to TDM1 pertuzumab. ADCs with payloads can affect bystander cells and may have greater utility in HER2 low or heterogeneous tumors. So moving on to triple-negative breast cancer. Triple-negative breast cancer is sort of the mainstay of treatment of cytotoxic chemotherapies. Now we have some antibody drug conjugates recently approved, and and immunotherapy, of course, but it still is a major issue. And so one concept that has been really discussed, has been considered for a long time, is can we differentiate triple-negative breast cancers into something that becomes more targetable or less aggressive. And so one study I wanted to highlight I thought was kind of clever. They used a reporter which was turned the cells green in the presence of estrogen receptor signaling. This was in a cell line, but then they were able to use thousands, about 10,000 compounds, and then looked for cells that became green, suggesting that they were now estrogen receptor was re-expressed and was able to signal in some way. And so this is showing the Western blot. It turned out that multiple kinases or multiple inhibitors targeted the same kinase, which was PLK. And so this is an example that if you pre-treat these triple-negative breast cancer cells with one of these PLK inhibitors, that in fact this made the tumors or greatly diminished tumor formation, and suggests that perhaps this could also be achieved in vivo. Can we sort of revert triple-negative to a more luminal like breast cancer? All right, and that's sort of the summary here. Okay, and so now moving on to technologies, there's great advances in multiple areas. I think one of the things that that academia and also pharma struggles with is how do we improve screening platforms to identify the right drug for the right patient? How do we continue to personalize therapies? So I just want to point out sort of a really impactful, I think, study that showed the power of using patient-derived xenograft models. And so this was a study where patients had their tumors biopsied, patient-derived xenografts, which are transplanted directly into mice, and then also patient-derived organoids were then generated from the PDX models. There was extensive molecular and phenotypic characterization, and then these organoids could be used for drug screens. So the advantage of PDX models is that they've shown to retain the molecular features frequently of the primary breast cancer, and also they retain the heterogeneity. So this was done, and as a proof of concept, it was shown that for a particular patient, you could predict a drug combination that went on to have substantial activity for this patient. Now, the challenge, of course, is that not all breast cancers can actually be propagated in this way. Only about 30% are actually capable of giving rise to PDX tumor models, and in particular, this is enriched in more high-grade tumors and also ER-negative tumors. So that still poses a challenge of how do we make personalized decisions for ER-positive cancers. This is obviously an area of a lot of interest, and I just wanted to show a study that tried to address this, which is using ex vivo patient-derived explants. So this is actual tumor that's grown ex vivo for a short period of time, on the order of about a week or so. And the study authors showed that, in fact, you can retain expression of estrogen receptor in these explants, because one of the challenges has been that as soon as you take breast tissue from luminal cancers and you grow it ex vivo, they often lose estrogen receptor signaling, and they sort of de-differentiate in a way. But in any case, they discovered that combinations of extracellular matrix combined with signaling inhibitors can allow you to grow ER-positive, or not grow, but maintain them in culture. Potential advantages are that you also have the immune repertoire present in these tumors, at least for a period of time. And so I think it remains to be determined if these can be used in some sort of high-throughput screen. But the clear implications here are that you take a tumor directly and grow it ex vivo. Moving on, I think another important question is, how do we identify synergistic drug combinations? There's far too many drugs And it's impossible to do this all in a phase 2 or phase 3 clinical trial. So one really heroic effort was to, in this manuscript that looked at 125 cancer cell lines, so 51 of them were breast cancer cell lines, and looked at 2,000 drug combinations with a total of over 100,000 drug responses that were identified. And so this allowed sort of pairwise comparison of multiple drugs. There's a lot of data in this, but looking just at the basal breast cancers, they were able to identify a series of different drug combinations that appeared to be synergistic. In particular, the BCL2 antagonists, such as navitoclax, seemed to synergize with multiple other drugs, as well as CHEK1 inhibitors. Unfortunately, some of these drugs, I'm not sure to what extent they're still in clinical development, but perhaps this would suggest that we should revisit them. So about 5.2% of combinations showed synergy, and navitoclax was particularly showed benefit in terms of synergy with multiple drug combination in triple-negative breast cancer and ER-positive breast cancers. Other technologies that I thought were innovative, this idea that, we talked at the beginning of the meeting about machine language or AI, and this concept that we could use imaging of cells in a dish to identify better combinations. This was a nice study that looked at high-content imaging called cell painting, using 6 immunofluorescent markers, and then it could, from this, extract 1,500 cell features. And then these cell features could be compared to genetic analysis or drug screens. So, for example, the imaging of perturbation of CDK2 was similar to treating with a drug called provalinol, which happens to also inhibit CDK2. So you can do these sorts of analysis to try to identify other drug combinations and doesn't rely on sort of classical screens, which really look just at cell death or look at ATP concentrations, for example. Okay, and so moving on, there are multiple studies using single-cell omic analysis. We've heard several really great sessions, and as I mentioned already, we now think of breast cancer as really kind of a heterogeneous disease, so it makes sense to incorporate some of this heterogeneity in our discovery and ultimately, hopefully, in our clinical translation. One study that stood out to me, really a tour de force, was not in breast cancer per se, but really trying to understand the normal breast. And so this stood out because it really used multi-omic approaches, including single-cell RNA-seq, CyTOF, which is a multiplexed fax-based type of analysis, cyclic immunofluorescence, and then also organoids. And this was from patients who had either had reduction mammoplasty or prophylactic mastectomy for BRCA mutation carrier. And so it actually led to a greater understanding of what are the cell types present within the breast, and also how do they change with aging, for example, or in the context of these high-risk underlying genetic alterations. Basically, it showed that there's spatially distinct epithelial subsets that are linked with age parity and BRCA2 status. In particular, there was this subtype, these alveolar cells that had poor transcriptional lineage fidelity and seemed to accumulate with age. They were thought to be alveolar cells, but they looked more like basal cells, and I think it remains to be determined, but the suggestion is that these might give rise to cancer in patients. Moving on to immuno-oncology, so I think the promise of immunotherapy has been that patients with advanced disease could be treated and not only have short-term responses, but perhaps durable remissions, which is seen in patients with smoking-associated lung cancer and melanoma. Unfortunately, that hasn't been the case to date. So, I think this is a very active area of investigation, trying to understand what's different about breast cancer, what are the evasion mechanisms So a couple interesting studies, one suggesting that you can actually have these transient cell-in-cell interactions where one cell will engulf another cell, and this renders the external cell sensitive to T cell killing, but the internal cell is resistant. And so you can see this, for example, here. And this study was both in melanoma, but also in breast cancer. And these cells were increased upon treatment with IO treatments. So T cells are excluded from killing tumor cells within other cells. And I think it remains to be seen, to what extent this really happens in patients, but it's certainly kind of a novel thought about how to evade the immune response. Another study explored dormant cells. So this is using engineered cells where you could determine which cells were dormant because they expressed a certain cell cycle inhibitor. And the finding was that there's quiescent cancer cells may actually form clusters together that exclude immune infiltrates and lead to increased exhausted T cells are found in this niche that are no longer able to kill these dormant cells. And what about in proliferating cells? So we usually think of biomarkers of immune response in terms of PD-L1 expression or MSI in terms of neoantigen production, but there's actual specific cancer genes which can lead to immune evasion. And so there are a couple of papers that explored the connection between one of the canonical oncogenes called MYC, which is frequently overexpressed in triple-negative and luminal B breast cancers, and found that there was an inverse relationship between MYC and inflammatory signaling, including MHC class 1 expression across multiple data sets, including the TONIC and TCGA and Metabric. A very similar paper looked at MYC oncogene and then downstream signaling through the CGAS STING signaling pathway. This is another inflammatory pathway that leads to interferon activation. And so what do we do with this information? The authors in both of these studies, I'm showing them side by side, showed that in animal models, PD-L1 was really not active, but you can use combination immunotherapy now with PD-L1 and then a TLR9 agonist and also an OX40 agonist, and now 75% of tumors were completely regressed and resistant to subsequent re-challenge with triple-negative breast cancers. In this study, using a MYC inhibitor plus PD-L1 also gave extensive or increased survival of mice. So the idea that targeting the MYC pathway or overdriving the immune response in these cells might be one approach to improve therapy. So what other immune components beyond T cells might contribute to the IO response? This is a very nice study that identified a specific subset of macrophages. So again, using single cell and spatial resolved analysis of heterogeneity, found that there's a particular subtype of macrophages that are perivascular that are found, and they're associated with tumor infiltrating CD8 positive T cells, and these are positively correlated with T cell infiltration and better prognosis for patients. Okay, and a hot topic over the last year was this question of metabolic requirements. So moving on to metastases, what are the metabolic requirements? So the question is, we know a fair bit about the primary tumor, but once tumor cells have spread to other sites, do they still have the same requirements? And this has been sort of a challenging question to address, but it's sort of been taken up by a number of different groups. One is a transcriptional approach that was published earlier this year, which looked at triple-negative breast cancers and tried to look at metabolic networks. This study was strictly transcriptomic, so I think it remains to be determined whether the transcription metabolic genes really translates into changes in metabolism overall. But what they found is that transcriptional changes of the metabolic network was kind of a hybrid between the primary tumor and then also the site of metastases. So it's somewhat variable, but for example, if you metastasize to the lung or to another site, you're also going to have different metabolism, which is related to the tissue of metastases. And so specific metabolic inhibitors may have utility against specific sites of metastatic dissemination. Another study that I thought was very excellent was to approach this idea of metabolism and metastases by asking a simple question of where don't tumor cells metastasize. It turns out that it's very rare to have metastases to skeletal muscle. We almost never see that. This study really tracked down that what seemed to be happening is that skeletal muscle is a highly oxidative stress inducing tissue, and basically while tumor cells can metastasize there, they just don't survive. If you introduce a gene that allows tumor cells to detoxify, basically increased oxidative stress, then now they can readily metastasize to skeletal muscle. So again, it suggests that the metabolism at different sites may actually regulate whether tumor cells can go to different areas. Other studies examined the determinants of the metastatic niche. And so this was a very nice study that was actually presented here, I think, in a couple of different sessions. So I'm not going to go into it in great detail. But one question is really, how does the extracellular matrix affect dormancy of disseminated tumor cells? And the key result was that a specific type of collagen, a type 3 collagen, seems to be important for keeping disseminated tumor cells dormant. Okay, and then finally, we've talked about, or we had several sessions talking about the microbiome, and so this idea that our bodies have lots of bacteria and fungi and all sorts of other microorganisms, potentially both inside and outside us, and perhaps in the breast ducts and so forth. But so how do these actually impact metastases? And so there's been a lot of work in this area over the last few years in particular, but a lot of it has been somewhat correlative, where we see that a certain bacteria might be associated with certain biological outcomes. So what I found interesting about this particular study is that they not only observed that there were intracellular bacteria, but that these actually had a biological function. They seemed to reorganize the actin cytoskeleton within circulating tumor cells, and suggest that they actually promote circulating tumor cell survival by enhancing the resistance to mechanical stress. So here we can link it to a specific biological function. And not to be outdone, there's also fungus among us, and so this was a pan-cancer microbiome atlas that was largely sequencing across 35 cancer types in over 17,000 samples across 4 different international cohorts that found, breast, but other cancers as well, often have fungus present within the tumor. So this, for example, staining of a breast cancer for aspergillus proteins, you could see that it stained positive, and then different types of fungi were associated with worse outcome. So multiple fungal bacterial immune ecologies were detected across tumors, and so intratumoral fungi stratified clinical outcomes, including immunotherapy responses, and perhaps cell-free fungal DNA might be helpful to diagnose healthy and cancer patients at early stages. And then finally, to leave you with this interesting and somewhat terrifying study, this paper that came out very recently showing that the circadian rhythm actually can affect circulating tumor cells. This is it's known that there's different genes depending on whether we're in an active phase of our day or whether we're resting. And so this study looked both in patients and in mouse models and found that during those resting phase there was much more release of these circulating tumor cells. And what they found was that there's a number of hormones, or the hypothesis is there are a number of hormones that are associated with these different phases that are regulated by circadian rhythm, and some of these could be manipulated, for example, giving dexamethasone to alter the number of CTCs. So I think stay tuned, but I think this is very provocative, and also, yeah, I'm not really sure what you do with this information, but it would suggest that during the rest phase, so while we're asleep, there's actually more disseminated tumor cells moving around. They also show that they're more functional, and they tend to set up shop in other sites. So in summary, there's lots of new basic research and technologies that have been identifying promising new mechanism, therapeutic strategies for different breast cancer subtypes. I think we're developing a better understanding of immunotherapy resistance mechanisms, and hopefully these can be integrated into future clinical trials. And I think, metastases is still the number one problem, where, we can't cure most patients with metastases, but I think we're understanding these vulnerabilities and mechanisms of dormancy are very important, and hopefully will lead to metastases-specific therapies. So thank you very much.